Okay. Oh, now it hates me. Oh, he's got a funky slide. Oh, crap. There we go. Okay, so this is basically what we just saw the video of. You can see that we've got our, let's see what order this goes in. Starts in the cell in the lower left-hand corner. You've got in the cell nucleus, the RNA polymerase transcribes the RNA from DNA. And we've got our piece of mRNA, which then travels out of the cell through the nuclear pores. It's got its cap on one end, and then it's got that tail that stuff can kind of fall off or get chewed off the end of. The large ribosomal subunit and the small ribosomal subunit, they come together and they clamp onto the mRNA, and they begin reading it. So it is binding to the mRNA. Then the tRNA molecules become attached in those specific places. So we've got our codon and our anticodon, and they're attaching to one another. And the amino acids are coming in, and they're forming this big long chain. And then the tRNA floats back off and into the cytoplasm on the outside of the nucleus. We've got these long chains of amino acids that make up our proteins. So we have our, in the upper left-hand corner, the polypeptide chain grows until the protein is completed. So polypeptide chain is all of those smaller subunits of amino acids, and they are growing until they make the protein that they are designed to make. Let's see, phosphorylation or other chemical modifications can alter the activity of a protein after it's translated. I'm not sure exactly what he means by that. So turning genes on and off. Usually we think of our genes as something that's always on, but genes can actually be turned on and off at specific times in our lives. So if you think back to what the baby looks like as it's growing inside of its mother, it may have specific things like your tailbone starts to grow, and if you don't have that gene turned off, then you can actually have babies that are born with tails. And if you Google that, that's really a fun Google image search of babies born with tails. And there's a really interesting video that I'll show you guys here in a minute about turning genes on and off. So the genes are typically controlled at the level of transcription. So transcription is before translation. In prokaryotes, so prokaryotes are our bacteria, proteins either block or allow the RNA polymerase access to the promoter. So if they are repressors, it means the gene is off. So in prokaryotes, the bacteria may have proteins that turn those genes off, or they may have activators which make the promoter more accessible and turn the genes on. So repressors off, activators on. So most genes are turned off unless they are needed. So most of the time the genes are going to be found in the off position unless they're needed to do something very, very specific. So here is an example of the lac operon. And it says the operon is a group of several genes along with the elements that control their expression as a unit, all within one section of DNA. Some sizes of these elements are not shown to scale. So the lac operon is one type of gene. And so in the first picture on the left, you see the promoter. That is when it recognizes and binds to the mRNA. The operator is the region where a repressor protein can bind to it to block the RNA polymerase. So basically, the repressor protein stops that whole process. And so if you look at The genes, we have 3 genes on that strand of DNA, and whenever the RNA polymerase runs into a repressor gene, it turns off the protein production, and then whenever it runs into an activator gene, it turns on the protein production. So here it is in action. Remember, we're talking about bacteria here. So in a bacterium E. coli, and E. coli are what are commonly found in your colon in the human gut. E. coli, the glucose is its preferred source of food, but it can utilize lactose as a backup. So usually it likes sugar, but if milk sugars are available, it will use the lactose, the milk sugars, as a backup. The lac operon makes that possible for bacteria to not waste energy transcribing the 3 genes necessary for lactose absorption unless glucose is absent and lactose is actually present. So why would it produce the genes to use lactose if there's no lactose available? It prefers glucose, and so it uses glucose first. If the only thing left over then is lactose and it still needs energy, which all cells need energy to survive, then it will actually begin producing this lac operon, and it turns on the genes in the absence of glucose. So normally, if lactose is not present, the operon is off. So it's not making the necessary lactose absorption and digestion because it's normally not there. And then you see down at the bottom, if the lactose is present, the operon turns on, and it's slightly modified to bind to the repressor protein, preventing it from binding to the operator. RNA polymerase can then bind to the promoter, transcribes the genes necessary for lactose metabolism. So if lactose is present, if glucose is present, it uses glucose first. If there's no glucose for energy, then if lactose is present, it turns on the genes to use the lactose. So stop that one.